We're very happy and excited to welcome Mollie Huber. Originally, she was at University of Florida Diabetes Institute, where she published her recent paper in the lab of Ed Phelps, but now she's in Boston enjoying the snow and rain at the Joslin Diabetes Center. And her talk today is beta cell dysfunction during type 1 diabetes pathogenesis. And just a little bit about her. She completed her PhD in the biomedical sciences at University of Florida under the mentorship of Drs. Clayton Matthews, Mark Atkinson, and Ed Phelps. Her work applied live pancreas tissue slices generated from human organ donor tissue to simultaneously assess beta cell function and immune cell activity during the different stages of type 1 diabetes pathogenesis. And she's now a postdoctoral researcher at the Joslin Diabetes Center in the lab of Rohit Kulkarni. Her most recent paper, we're going to drop that in the chat in a second, and feel free to drop questions in the chat during the talk. Thank you so much for joining us, Mollie. We're excited to hear you walk us through this new paper.
Thank you. And then also with questions, people feel free to interject if there's questions during the talk too, because
great. It's good to know that.
Yes, it can be easier just to answer it then than jumping back and forth.
Got it. Yeah, I've got this in the, if I see anything in the chat, I'll stop you.
Okay. And so, hello everyone. Today I'll be telling you about the work I did during my PhD at the University of Florida on beta cell dysfunction, and that we investigated using the live pancreas tissue slice model. I can figure out how to progress. There it goes.
There you go.
Progress my slides. And so I think as most of us know, type 1 diabetes is an autoimmune disease characterized by the loss of the insulin-producing beta cells of the pancreas. People start out having a genetic predisposition to developing this disease. As kind of time goes on, and some of these people with this predisposition, you start to see autoimmunity, autoantibodies against islet antigens can be detected in the blood. However, they're still pre-symptomatic. As this autoimmune process continues, people become symptomatic, and this is when they're then diagnosed with type 1 diabetes and have a lifelong reliance on exogenous insulin. And so with this project, what we really wanted to focus on is what's going on within the pancreas during these early pre-symptomatic stages of type 1, because it's still very unknown, since the pancreas is a hard to access organ, and it can be difficult to determine what's going on within the tissues during these early stages of disease. And so it's been established for decades that there's this period of beta cell dysfunction prior to type 1 diabetes diagnosis. And so in this study published in 2016, children that had a high genetic risk for developing type 1 diabetes were followed from birth. And IV glucose tolerance tests were performed on them over time so that their beta cell function could be tracked longitudinal. And so with these graphs, the dotted line represents kids that did not progress to developing type 1 diabetes, and the solid line represents children that did. And what I want to emphasize here is when you look at their first phase insulin response following these IV glucose tolerance tests in the kids that did progress to developing type 1 diabetes, years before they're diagnosed, you're starting to see it decrease. And can everyone see my mouse, see what I'm pointing to? I think so. And so, yes. Okay. Also, when you look at the area under the curve of their insulin response, again, in this first phase, 0 to 10, or in this 0 to 10 minute range, about a year before they're diagnosed, again, you're seeing there's this decrease in this insulin under the curve. And so what this is showing is that while these people are not symptomatic and they don't have type 1 yet, their beta cells are starting to become dysfunctional up to years before they're diagnosed. And so there's this dysfunctional period prior to type 1 diabetes diagnosis. And so one of the major models used to study type 1 diabetes are isolated islets. And we've learned a lot of information from this model. And so how this works is donor pancreas is received, it's infused with enzymes. The pancreas will then be cut into pieces, digested. There will be a gradient separation, and then you have your isolated islets. And so this is great because it allows you to do functional studies to see how the islets are functioning at these different stages of disease progress. However, there are some drawbacks to this methodology. During the isolation procedure, the islets are removed from their surrounding microenvironment, which means you lose a lot of the underlying pathologies that the islet is existing in, particularly in the context of type 1 diabetes. Also, because this is a very kind of rough procedure for the islets and you get to the cutting of the pancreas and the enzymatic digestion, islets that are already fragile, particularly insulitic islets in the context of type 1 diabetes, are not able to survive this procedure. And so while we still are learning a lot using the isolated islet model, you have to remember these drawbacks, and particularly in the context of the type of the insulitic islets having trouble surviving this procedure. And so we can use a lot of different models and methodologies to study type 1 diabetes at kind of different levels of tissue development. And so you can look at things kind of systemically with the systemic metabolism. This is like the IV glucose tolerance test done with the children mentioned in the earlier figure. Pancreas tissue slices allow you to look at things within the organ tissue environment. Isolated islets allow us to see the endocrine functional unit, and then single cells allow you to really assess cell biology. And so for this talk, again, I'm going to be mostly focusing on the pancreas tissue slices, which allow us to look at the islets while in the organ tissue environment. And so the need that this work seeks to address is that previous methods have been unable to observe intact live pancreas tissue and its related immune cell interactions in the context of type 1 diabetes. And the objective is to assess islet functionality at different stages of type 1 diabetes development to determine the impact of these beta cell immune cell interactions. In addition to isolated islets, histology and fixed tissue staining has also frequently been used to study type 1 diabetes in both mouse and human tissue. So here you can see kind of the autoimmune process happening within a mouse, where you can see a large amount of immune cell infiltration, these later disease stages. And you're also able to see the T cell infiltration within human tissue shown here into these islets indicated by glucagon staining in red. And so while with histology, you're able to see what's going on within the pancreas, because the islet is maintained in its surrounding microenvironment, the tissue has to be killed during the fixation procedure. And so you're only getting a snapshot as to what's going on in that organ at that specific point in time. And you're not able to look at what's going on functionally within the islet or the surrounding tissues or the immune cell interactions. And so this brings us to the slices, where they are alive and functional, and we can do different functional assessments using this model. And so this is data that was acquired by NPOD, where they do perfusion studies. And so they'll put multiple slices in a chamber and perfuse different solutions over them, collect the effluent, and then do an insulin ELISA to quantify how much insulin is produced in response to these different stimulations. And so this is very useful because it allows us to look at how all of the beta cells within multiple slices are functioning. So this kind of gives us a systemic functional profile for every case that comes in. And so you can see representative insulin traces from some of these cases up here. This donor with no diabetes has the expected insulin response in response to high glucose and potassium chloride. We see a similar trend with the autoantibody positive donor. We again see the response to high glucose and potassium chloride. However, when we look at this trace from a donor with type 1 diabetes, we still see that they have a potassium chloride response, so it is blunted, and there is no longer a response to high glucose stimulation, whereas we would expect there to be a peak about halfway at this peak in response to the high glucose stimulation. And so what this is showing is that there are still beta cells present within this donor. However, they are no longer responding appropriately to high glucose stimulation. This is emphasized further, and we start to quantify these in a number of cases by looking at the area under the curve per minute during these different stimulations with different donor types. And so donors with no diabetes have the expected insulin response in response to high glucose and potassium chloride. That trend is maintained in donors that are autoantibody positive. However, when we look at donors with type 1 diabetes, we see that this insulin response is lost. We can look at this in a little bit of a different way by splitting it out by stimulation type, whereas the basal activity is the same between these three donor groups, and that they're not producing insulin in response to low glucose as expected. However, when we look at the high glucose stimulation, donors with type 1 diabetes have a diminished response to high glucose, and that they're not producing the appropriate amount of insulin. And finally, when you look at the KCL stimulation, there's no significant difference with that as well between the groups. They're still responding to potassium chloride, but they're not responding to high glucose challenges, indicating a beta cell functional defect. And so the overall question that we're seeking to address with this work is, what are the functional and molecular changes that occur in beta cells prior to type 1 diabetes diagnosis? And so we hypothesized that dysfunctional glucose responses in diabetes occur due to alterations in the glucose metabolism pathway. So to kind of give an overview as to the different experiments and methods we're going to be talking about in this paper, the schematic here. And so NPOD will receive a human organ donor pancreas. They'll take different sections of tissue from the tail and then process it in some different ways. And so one set of generated slices will be used for the perfusion studies that I was talking about earlier, that again kind of gives us a systemic profile as to how the beta cells are functioning within multiple slices, and we can look at how much insulin is produced. Another set of slices that we receive will perform microscopy experiments on to look at how the beta cells are functioning within the slice. And so how we do this is we put the slice on a confocal microscope in a special chamber. We use syringe pumps to stimulate the slice with different stimulations, so low glucose, high glucose, and potassium chloride. And then we can then look at how the fluorescence changes, particularly with the calcium indicator, to see how the beta cells are functioning. And with the microscope as well, we're also able to simultaneously look at immune cell activity, particularly T cell activity within the slices. Because with the live slice studies, time is always a big factor in that you only have about 3 or 4 days before the slices have a big drop in viability. We also did a fixed tissue staining of additional beta cell glucose metabolism markers that allow us to dive in more mechanistically as to where this dysfunction could be occurring within the beta cell. And then these fixed tissue sections are also from NPOD. So again, a major benefit of the slice model is that it allows the tissue to remain viable, functional, and also to maintain its endogenous pathologies. So to give an overview as to how this slice generation works, again, NPOD will receive a pancreas. They will cut small piece sections of tissue about 5 millimeters by 5 millimeters. They will embed these tissue sections into agarose and cut them using a vibratome. And then that will give us the 120 micron thick tissue slices that you can see here. To give you a better idea of what a slice looks like, you can see a dark field stereo microscopy image of one here with the islets outlined in white. As you can see, the islets make up a fairly small percentage of the tissue overall. Due to the granularity of the insulin-containing beta cells, we can use reflected light to locate islets within the slices as well. And you can see islets kind of lighting up using that method here, indicated by the white arrows. As I mentioned before, one of the major benefits of the slice model is that the islet is maintained in its kind of surrounding microenvironment. And this is emphasized by this laminin and collagen 4 staining here, where you can see an islet outlined in yellow. We also are able to take 3-dimensional Z-stacks of islets within the slice and can make out individual cells within them, and that's shown here with this kind of rotating image. So now that I've explained the slice model a little bit, I want to now get into the beta cell kind of glucose metabolism question that we have. So as I mentioned before, it's been well established that there's this period of beta cell dysfunction prior to death in type 1 diabetes. And so this is emphasized by this study that was published in 1984, where 4 children that either were at very high genetic risk for developing type 1 or that had recently developed type 1 were given different IV tolerance tests to see what their insulin responses would be. And so when they were given IV glucose, which again has to be metabolized through this entire pathway for insulin to be secreted, they produced little to no insulin. It was effectively no insulin. So that means something in this pathway is going wrong, or that the beta cells are dead. However, when they were given IV arginine, they were still able to produce insulin. And so that shows that the beta cells are not dead, they're alive, they're just not responding appropriately to this glucose challenge. So then the question becomes, where in this pathway with the IV arginine acting at this latter point, so prior to that, where in this pathway is this beta cell dysfunction occurring? And so we use these slice studies to try to address this question. General experiment overview when we receive these slices is that we will incubate them for at least an hour at 37 degrees Celsius to get them up to body temp. We will then incubate these slices and kind of our low glucose KRBH buffer and stain them with functionality indicators. So in our case, the calcium indicator, immune cell antibodies, typically the T cell antibody, and also Cytox blue to assess viability. And then we will then perform dynamic functionality experiments and immune cell imaging simultaneously on these tissues. So to start, as I mentioned before, you can use reflectance to locate islets within the slices, and also that the islets make up a fairly small percentage of the slice overall. However, we're not just interested in the islets, we're interested specifically in the beta cells. And so we wanted to have a surface marker that would allow us to identify beta cells for live tissue studies. And so we use this ENTPD3 marker that was previously published by the Saunders group. And so ENTPD3 is a surface marker that's present on beta cells, and it's been shown to work in, I think, fixed tissue and isolated islets. But we wanted to verify that it would work for our purposes in these slices. And so how we did this is within a slice, you can see an islet located using reflectance here, and we stained with ENTPD3. You can see it's staining the same regions. To verify that it did indeed stain beta cells within these tissues, we took fixed tissue and stained with ENTPD3, insulin, and glucagon. And when we did that, we found that the ENTPD3 shown in yellow co-localizes with the insulin shown in red, and it does not co-localize with the glucagon shown in blue. And we verified this both in donor tissues from a donor with no diabetes and also from a donor with type 1 diabetes. And so we made sure that the ENTPD3 would work for us within the slices. We next wanted to see that it could be applied for beta cell identification in calcium recordings, which is our major functional output in these studies. And so you can see these top images from a donor with no diabetes, bottom images are from a donor with type 1 diabetes, and they're from calcium recordings. And so you can see the little green pop out is shown off to the right. You can see beta cells identified using ENTPD3 staining and regions of interest drawn over those beta cells in the calcium channel for the calcium recording. And when we look at the changes in fluorescence due to these within these specific cells here during different stimulations, we see the expected increase in fluorescence in response to high glucose and potassium chloride within the donor with no diabetes, and a loss of the high glucose response in response to a loss of the high glucose response in a donor with type 1 diabetes and a maintenance of the potassium chloride response. This trend is emphasized further when you look at heat maps of different recordings within these different donor groups. Every heat map is a single recording. You can see the case off to the side. Every row on the heat map represents an individual beta cell within that recording. And then as the heat map transitions from blue to green to orange, that is the cell getting brighter and increasing in fluorescence in the calcium channel. And so when you look at these functional profiles in these different recordings, we can see donors with no diabetes have the expected increase in fluorescence in response to both high glucose and potassium chloride. There's a similar trend with the autoantibody positive donors, where again, we see the high glucose and potassium chloride responses. However, when we look at donors with type 1 diabetes, we see a loss of the high glucose response and a maintenance of the potassium chloride response. And so what this is showing is that these beta cells are alive and they're still able to respond to stimuli because they're responding to potassium chloride. However, they are no longer responding appropriately to high glucose stimulations. This trend is emphasized further when we look at traces from all of the recordings that we took through the course of these studies, where again, when we look at donors with no diabetes, we see this increase in fluorescence in response to high glucose and potassium chloride. Same trend with donors with autoantibody that are autoantibody positive. However, when we look at donors with type 1 diabetes, again, they lose this response to high glucose and they maintain their response to potassium chloride. Again, emphasizing it's beta cell dysfunction, not just beta cell death. I'm going to give you an idea of what these calcium recordings look like. I have some representative videos here. So again, to do this, we take the slice, we put it on a special chamber on the microscope, and we perfuse different solutions over to record the response of the beta cells.
Can I just ask you a quick question? Is this special chamber, is this something devised at UF or is this something commercially available?
No, the chamber is commercially available.
Great.
And it should be in the
Methods. Yeah.
not
Okay,
just
great.
shoot me an
I
email.
might have missed it. Great. Thank you.
No, it's commercially available. And so the reason that we're looking at calcium is just prior to insulin being secreted, beta cells increase their intracellular calcium. And so how that looks in this recording is these cells will turn a brighter green. And so that's how we're assessing beta cell function within these recordings. And so you put the slice in the chamber, you perfuse different solutions over it, and then you see how the fluorescence changes in response to those different stimulations. And so here you can see beta cells that was stained with ENTPD3 shown in yellow, T cells shown in magenta, and the calcium indicator in green. And so these top set of recordings are from a donor with no diabetes. So we're expecting things to kind of function as they should. You can see basal activity in the flashing of beta cells in the low glucose solution. You switch to high, you can see the beta cells light up in response to that high glucose stimulation. So again, things are functioning as they should be. And then we stimulate with potassium chloride. Again, the beta cells light up in response. And so again, these recordings from a donor with no diabetes, everything is functioning as we would expect. This bottom set of recordings is from a donor that was diagnosed with type 1 diabetes at onset, and this islet is also fairly infiltrated by T cells. And so when the islet in this recording is in low glucose, we see that basal activity, so these beta cells are alive. However, when we stimulate with high glucose, they all kind of stay at that basal level of activity. They're not lighting up like they did in that above recording. So they're not responding appropriately to this high glucose challenge. However, when we stimulate with potassium chloride, they do light up in response. And so what this is showing us, again, is that these beta cells are alive. They have basal activity. They're able to respond to stimuli because they're responding appropriately to the potassium chloride stimuli. However, they're dysfunctional in that they are no longer responding to the high glucose stimulations. And so we quantified these recordings a couple of different ways. And so one of the which is to look at the peak calcium response achieved during these different stimulations. So 3G is low glucose, 16.7G is high glucose, KCl is potassium chloride. And when we look at these different donor types, every dot being a recording and every color, different color shade being a donor, what we can see is there's no significant difference in the basal activity, so at low glucose, and there's no significant difference between the donor groups when we look at the potassium chloride response. So kind of at the upper end response of these cells. However, when we look at the high glucose response, donors with type 1 diabetes have a significantly diminished response to high glucose. So it's just showing that beta cells are dysfunctional and they're not responding appropriately to high glucose. And we see this same trend when we look at these values different ways. When we take the ratio of the high glucose response to the low glucose response, again, donors with type 1 diabetes have a significantly diminished response. Additionally, when we look at the fraction of responding beta cells within these islets, so say there's 10 beta cells in a recording and only 5 respond, that would be at the 0.5 level on this graphic. We see that in donors with type 1 diabetes, a minority to none of their beta cells respond to high glucose stimulation. And so this is significantly diminished compared to both donors with no diabetes and autoantibody positive donors. And so in addition to looking at functional information with the slices, we're also able to kind of see different tissue phenotypes at these different stages of disease development within the live pancreas tissue slice model. And so these are all just stills from recordings that we have. And so in donors with no diabetes, typically islets kind of have that round regular shape. You can see the beta cells in yellow. There's not a lot of immune cell activity around them. With autoantibody positive donors, typically we also don't see a lot of immune cell activity. This one is a little bit higher than what we would usually see with the T cells shown in magenta. But again, the islets usually look okay. And then when we get to donors with type 1 diabetes, we can see very large amounts of T cell infiltration occurring. And it's not uniform. Sometimes there's a lot of T cells, and sometimes there aren't many within donors that have type 1 diabetes. So we're able to see kind of how the tissue looks at these different stages of disease development within this slice model. So in addition to the calcium recordings, we were also able to record live endogenous human immune cell interactions with beta cells kind of endogenously within these slices. And so here you can see again, the beta cells are shown in yellow, T cells are shown in magenta, and you can see them all kind of going in and interacting with the beta cells within these recordings. This one, it's that little X of beta cells, and here they're kind of coming in on the two different sides of the islet. Another interesting kind of aspect of this is that you can see heterogeneity even within the same case with the level of immune cell infiltration. And so all three of these images were taken from the same case. And so these two islets are very heavily infiltrated, and then this one is very minimally infiltrated, all from a donor with a recent onset type 1 diabetes. And so the phenotypes you see are not uniform even within the same case. And so
Molly, can I just ask a question? When you looked at these cases, did you see their response to glucose in the different islets?
yes.
What they differed, right? And we did talk briefly offline a little bit about sort of the Benninger work and how this whole idea of the leader beta cell or leader islets, I guess, or leader beta cells, can kind of could possibly drive insulin secretion. And then if those get, taken offline, what would you hypothesize? I mean, are you thinking that this could be something you're seeing?
So we will get to the T cell impact on beta cell dysfunction in a couple of slides. And
All
it's
right, great. Sorry to
very
preempt.
interesting.
I'm waiting. This is such beautiful work. It's really, really nice.
Thank you. Okay, so now we're still on the T cells in these recordings. So since we were able to record all of these like interactions and insulitis happening with this elevated T cell infiltration within these recordings, we wanted to quantify it to see how the T cell, if the T cell motility would change, whether the T cells were within the islet or outside of it. I want to give a shout out to a student I work with, Th1nk, who had to sit in a dark room and manually track all of these T cells in these videos. But it came out to be really interesting data. And so what we saw is that when the T cells are within the islet, they slow down. You can see here most of these tracks are blue, indicating slower tracks, versus red, indicating faster T cell motility. And when we kind of quantify this in different recordings, generally what we're seeing is that the T cells are slower in the islet than they are in the exocrine tissue, which this makes sense logically, but it was very nice that we could see it within these recordings that we were able to capture. So because we are able to look at the beta cell function through calcium recordings and also simultaneously look at what the T cells are doing within the slices, we wanted to put that together to see how is the amount of T cell infiltration relating to the beta cell dysfunction? Because it would make sense that there would be a larger amount of T cell infiltration and the beta cells would be dysfunctional. So we wanted to see if that would in fact be true. And so these are representative images from two different recordings from the same donor. This islet is highly infiltrated. This islet does not have infiltrating T cells. And what we see is that whether there are T cells there or not, the islets are not responding to high glucose, and they're still responding to KCl. And so this is showing that the beta cells are alive. However, they're not responding appropriately to the high glucose challenge, and that this effect does not seem to be being driven by T cell infiltration. This is emphasized further when we kind of quantify across all of our recordings. And we split out the donors with type 1 diabetes into recordings that have 0 to 5 infiltrating T cells, so minimally to non-infiltrated to highly infiltrated, in the recordings with 6 or more T cells. And what we see is that the beta cells are dysfunctional either way. Whether the T cells are there or not, the beta cells are not responding to high glucose stimulation. And so this was very interesting to us. We also wanted to double check that these really were cases with insulatus. And so we confirmed it by looking at both the NPOD histology database and also from immunofluorescence staining done by us to see that if we were really seeing insulatus in these tissues, and we were. And so again, what we found here is that beta cells are dysfunctional regardless of the amount of T cell infiltration. And so we next wanted to do is that if it's not the T cells kind of inducing this inflammation from what we were seeing, then the question becomes, what is the mechanism of beta cell dysfunction? So we investigated this a couple of different ways. Previous work conducted by the Matthews lab used laser capture microdissected islets from human tissue to look at how RNA expression changes at these different stages of disease development. So we use this to look at RNA expression of different beta cell glucose metabolism markers. In addition to the RNA expression, I also performed traditional immunofluorescence staining of 10 different antibody panels over 300 slides from donors at different stages of disease development. So no diabetes, single autoantibody positive, multiple autoantibody positive, and donors with type 1. We imaged around 30 islets per slide, except in some donors with type 1, there weren't 30 islets within the slide, in which case we would image all of the ones that were present, which meant that we had around 9,000 islet images to analyze to look at how protein expression was changing at these different stages of disease development in these different glucose metabolism markers. You can see some of the breakdown here of these different cohorts. Something I want to emphasize is that the RNA expression cohort and the protein expression cohort are different people. And so just wanted to point that out. And so to start, we looked at class 1 HLA, so kind of beta cell visibility to the immune system and also insulin. And so in these top graphs, we look at class 1 HLA at both the RNA level and at the protein level. We see it go up in type 1, which this makes sense. And interestingly, we look at insulin expression, particularly at the protein level as type 1 diabetes developments, we're not seeing that change at the different stages of disease development. And I may be saying, well, at the RNA level, it's going down. And so something I want to point out here and to keep in mind for the other markers we look at is that the RNA cohort is from laser capture microdissected islets. And so they cut out the entire islet from the tissue. And so you're not just getting beta cells with this, you're also getting alpha cells, just the different cell populations in the endocrine unit. And so it's not beta cell specific. So particularly in the context of type 1, if the general number of beta cells are decreasing within the islet, that would make sense for the decrease in the insulin. However, for the protein expression for that analysis, we were able to specifically mask on insulin. And so this allowed us to look at the expression on a per beta cell basis. And so what we're seeing is that individual beta cells have the same amount of insulin at the different stages of disease development. And what this means mechanistically is that the beta cell defect that we're seeing is not a result of the beta cells not having enough insulin. They still have insulin. They're just not behaving appropriately, and the dysfunction is occurring earlier in the pathway and they're not secreting it. And so when we get into different kind of markers and components of beta cell glucose metabolism that we investigated and that we saw were different, things start to get a little interesting. When you look at GLUT1, which is the glucose transporter, so it's how glucose gets into the beta cell, we see a decrease in type 1 diabetes at both the RNA and the protein level. Also, for the next few slides, the top graph with the name in italics is at the RNA level. The non-italicized lower graph is the protein expression. And we also can see this decrease in GLUT1 expression pretty clearly with these representative images as well. So glucose transporter being downregulated in type 1. PFKFB4, which is a regulator of this phosphofructokinase, which is a rate-limiting step in glycolysis. So very important for glucose metabolism. At the protein level, we're seeing it decrease in both multiple autoantibody positive donors as well as donors with type 1. So this is happening prior to diagnosis in the disease process. So before you see that large amount of T cell infiltration occur, you're already seeing some functional changes occurring in the beta cells, and that this phosphofructokinase is decreasing.
Just a quick shout out to the Marins and Kibbe counterpoint in diabetes, April 2024, where they proposed an alternative model based on spatial and bioenergetic specializations of the beta cell metabolism, in which glycolysis initiates insulin secretion. That was highly debated at the Sugar Science, as many of you know, but this is very interesting finding. And do you have anything to sort of say about speculate about how that might be dovetailing with their findings? You don't have to go out on a limb, but I did want to just kind of bring it up.
Yes, I think I like that I think it's good to look at the beta cell also being a problem in this process. I think that it's good to evaluate how beta cell it makes sense with how the disease progresses, that the beta cells are metabolically abnormal, particularly when you look at how you see those functional changes occur in people where their first phase insulin responses are decreasing years before they're diagnosed. And that's years before you see a lot of that widespread immune infiltration. So I think it makes sense to consider metabolic defects within the beta cell. And glycolysis is a big part of beta cell glucose metabolism. So
True.
Okay. So yes, PFKFB4. It decreases in both multiple autoantibody positive donors and donors with type 1. We also see GAPDH becomes upregulated within this pathway, again, in donors with type 1 diabetes. And so again, this is another component of glycolysis. We also see SDHC, so now we're getting into succinate dehydrogenase. It's involved in the citric acid cycle and the electron transport chain. And at the RNA level, it's decreased in donors with type 1. And at the protein level, it's decreased in both multiple autoantibody positive donors and donors with type 1 diabetes. So again, this is another critically kind of important component of the beta cell glucose metabolism pathway that we're seeing differences both in multiple autoantibody positive donors before disease diagnosis and also donors with type 1. KCNJ11, which is the potassium gated ion channel, goes down at the protein level in donors with type 1 diabetes. Interestingly, the calcium channel at the protein level we found to increase in donors with type 1. And so you could debate why this could be occurring. It could be the beta cell trying to compensate for other functional defects. We're not sure, but it was like a very clear trend that we saw where the calcium channel expression was increasing. And so all of this to say, we kind of have this beta cell glucose metabolism schematic here. Everything in blue is something that we stained for. And a heat map showing how the expression changes. If it is red, that means it's increasing compared to donors with no diabetes. Blue is less expression. And something I want to emphasize kind of when you zoom out here is the amount of blue in this half of the graph, which is the multiple autoantibody positive and T1D positive donors, versus this half of the graph, which is the no diabetes and single autoantibody positive. So kind of you can think of the disease process going this way. And a lot of these metabolic markers start to be downregulated, particularly those involved in glycolysis, the citric acid cycle, and oxidative phosphorylation. Another interesting thing I want to point out on this heat map here is that we looked at the ER stress markers, PERK and IRE1 alpha. And what we found is that they did not change in these donors at these different stages of disease development, which we thought was very interesting. So to conclude, what we found is that beta cell function is reduced during type 1 diabetes development, and this is indicated by a loss of the responses to high glucose stimulation. We also found that beta cell dysfunction is not driven by T cell infiltration, suggesting that it occurs prior to immune cell infiltration into the islets. We also saw alterations in the glucose metabolism pathway that indicate metabolic dysfunction that's occurring prior to disease onset. So this graphic here, you can see the arrows, and that indicates which way those markers were changing at these different stages of disease development. So this was a huge group effort. So I want to kind of go through some acknowledgements, particularly my mentors, my PhD supervisory committee, Phelps Lab, Thompson Labs, Spire and Slack Rupnik Labs, Matthews lab, the Atkinson lab, Nebraska lab, the NPOD slice team, who they come in middle of the night, weekends, holidays to provide access to these tissues and generate these samples for us to use. Special shout out to Alexandra and Adrian, who helped with all the data analysis that I talked about here. And I want to especially thank the NPOD donors and their families, because if they did not make the decision to donate, then none of this work would have been possible. So thank you, and let me know if you have any questions.
Thank you so much. Beautiful work, and really just a tremendous collaboration coming out of University of Florida. I wanted to just sort of tap one last thing, just sort of like bringing up the beautiful work of Jing Hughes at Wash U St. Louis. You know, they're looking at the primary cilia on the beta cells. I wondered if you guys noted any morphological changes in primary cilia corresponding to the metabolic changes you saw in the slices. I don't know if you can capture them very well in slices, but I just thought I'd throw it out there.
It is not something that we looked at, but that's like the struggle with slices, is there's always a lot more interesting things to look at than like what you are feasibly able to look at in the time that you have them.
And yeah, it's just kind of a curious, you know, there's been some smoking guns around that. The primary cilia, whether or not it can be impacting the functionality of either the leader beta cells or the beta cells at large. And then I guess I would also ask, you know, there's some buzz still around the alpha cells and their role in stressing basically the beta cells. Did you guys look there to see if there was any kind of interface between the two cell types in your construct?
We did not look at that specifically. You can see the alpha cells when you do the calcium recording, because when the beta cells turn off, they turn on, like they'll kind of act in an inverted way to each other. It's not something we specifically analyze, but we do have the recordings. Like, we want to make them available. So if there's things within them that other people want to look at, they can.
That's nice. That's really great. Very collaborative. Here's a question from Nathan Cook. Have you stained for beta cell maturity markers such as Th1nk in these GLUT1 downregulated glucose non-responsive T1D islets?
We have not. Yeah, so would be interesting. I would expect they would likely be mature because they're all from adults. They're primarily from adult samples, but that would be something to look at to see if there's anything odd going on with that.
Nice. Anand Hardikar, nice talk, Molly. Very nice body of work. I know you mentioned laser microdissected sections. Have you been interested in measuring microRNAs in these laser catapulted sections, captured sections in single cells?
So the micro dissection and the sequencing was done prior to me joining the lab, but I think if they're doing it at a single cell level and kind of looking at things more specifically would also be interesting to do down the line.
Great. Jean-Luc Tran, have you looked into cytokines PGE2 level from the pancreas?
No, we have not done that either to PEG2. We have not looked at the cytokine levels in these. Either, but I guess you could probably pull, if you wanted to look at that, you could pull media and see what they were secreting. But that was, again, something we did not look at with this cohort.
Yeah, it's hard to get everything done.
Yes.
And it does show that this system is kind of ripe for more inquiry, so that's exciting.
Yes.
All right, any more questions? Last call for questions. And are you gonna, my question, I guess, is now that you're up at the Joslin in your new lab, are you going to be furthering this system, or what's your new project?
We're
If you can
still,
speak on it.
yes. I'm looking, I love slices, but I'm looking to kind of diversify my skill set and learn to look at things from some new kind of facets and directions. So break from slices for right now, but we shall see.
Right. I'm sure you'll be, adding a lot of value to the knowledge base. So we're looking forward to your next steps. Thanks so much for joining us today. And we look forward to hearing from you in the future. Have a great rest of your day.